Okay, everybody. So now that you have learned the cellular organelles, now we're going to focus on the plasmamembrane and how it is able to allow certain things to enter and exit using its selective permeability. So things you're going to need to know in this section are basically understanding what the components of the cell membrane are and how it's able to then maintain the internal environment that it does in the cell. We're going to learn about the fluid mosaic model, and that's just kind of a function of how the membrane is organized. And then going through and understanding how if you change what is in the structure of the membrane, then that's going to affect its permeability. So again, that just goes back to the fundamental idea of changing structure changes function. And finally, the cell wall and how that relates to certain prokaryotic cells or plant cells in maintaining structure and function. Okay, so first up, we have membrane structure. Just going to click through this guy so that I can talk about it. But cell membranes, as I mentioned, is selectively permeable. If something is permeable, that means that something can go through it. But not just anything and everything can go across the plasma membrane. In this case, it's selective. So some substances are able to cross more easily than others. And we're going to find that some just freely float on by, others will need helpers to get through, and some actually even require energy to make it happen. And this goes hand in hand with the idea of the fluid mosaic model. The idea is that the plasma membrane is fluid. It has weak interactions that are holding it together so that, it is a solid structure, but there is a fluidity there. It is able to move a little. And that's what kind of gives animal cells a more of a relaxed structure, some movability about it. Mosaic means that it is composed of many different things. In this case, the structure of the membrane includes things like phospholipids, and as we will see, a number of proteins, carbohydrates, things like that. So it's not just a singular unit or, how we learned in macromolecules, a monomer building to a polymer, but it's really a collection of many different things together. So it's weak interactions giving it fluidity, and its diverse composition gives it its mosaic nature. So what is a phospholipid? Well, we learned macromolecules and we learned what lipids are. And we learned that lipids have long chains of hydrocarbons. In this case, phospholipids is one kind of category of lipid. We had triglycerides, we had steroids. Phospholipids are hit heavily here. And that is what the plasma membrane is actually made of. It's a bilayer. If you can see in the picture, there is a top row of phospholipids and a bottom row of phospholipids. And it organizes itself so that it's kind of two layers sandwiched together. And you can see the phospholipid structure. It has a phosphate head, and phosphate does have a charge associated with it. And then it has two hydrocarbon tails. And just like we learned with other lipids, it can be saturated or unsaturated, so they can be a straight chain. If saturated, it can have a kink in it if it's unsaturated. So the overall structure is considered amphipathic. Basically, it means that it has different properties on each end. The head group, because it has a phosphate attached to it, is hydrophilic, loving water. And the long hydrocarbon tails, as we know, are hydrophobic. They fear water. So because of that, the plasma membrane itself organizes so that the hydrophobic tails are hiding away from the water. That's why they kind of have it tail to tail. And the hydrophilic heads love water, so they're going to be on the outside. And that's why it forms in this bilayer structure. Overall, it keeps a lot of hydrophilic things from just floating on through because of that hydrophobic kind of gap in the middle there. Nothing that's hydrophilic is going to want to float its way through from one side to another. So here's an example of what that looks like in terms of starting to incorporate other things. The membrane itself, because the tails are saturated or unsaturated, there's a little bit of that fluidity there. So that's kind of what gives it its fluid nature, as well as the proteins that are inserted into it. So that's where the mosaic piece comes in. In terms of the protein, the hydrophobic parts of the protein are going to be towards the interior, which makes sense. It's hiding away from water. And the hydrophilic regions are going to be the ones on the surrounding portion, where they are more exposed to a water environment. And as we know, when proteins fold into their 3D structure, the tertiary part is where the R groups interact and form these proteins. Here's what it looks like in a more accurate picture. It's kind of a hot mess of a lot of different things. So the bottom is kind of the inside of the cell, and the top part represents what would be on the outside of the cell. So when it says cytoplasmic side, it's the side that the cytoplasm is living on, and then extracellular literally just means outside the cell. So you can see the bilayer made of phospholipids as here. There are tiny steroids, so each one of these little guys next to the phospholipid is cholesterol specifically. So you can see that kind of sprinkled in throughout the membrane, and that's going to change its overall fluidity. You can see that there are transmembrane proteins, ones that kind of go all the way across the membrane that are embedded in. They're also called integral proteins because they're integrated into the membrane. There are what we call peripheral proteins that we will learn about. And to be on the periphery or peripheral, think about peripheral vision. It means on the side. So peripheral proteins are located only on one side. In this case, the ones in this picture on the bottom there are only on the cytoplasmic side. This is contrasting the integral ones that literally are integrated and go across both sides. So the peripheral proteins can be stuck on the inside or they can be stuck on the outside too. And you can see on the outside are glycolipid chains. So there's all kinds of extra things, carbs, fibers, all kinds of stuff that can connect to the outer portion of a cell. Okay, so in terms of membrane fluidity, like I mentioned, the tails, as we've learned, we know the difference between saturated and unsaturated. So those kinks, just like in, triglycerides, and when they are unsaturated, we get liquid at room temperature versus when it's saturated and they're solid at room temperature. That happens here. The unsaturated nature of these phospholipid tails prevents super close packing. So that gives it an increased fluidity. If it does become more saturated, then it's going to become thicker and stronger, and then kind of goes more towards solidification. Cholesterol, in its ability, is able to kind of resist change. Cholesterol reduces fluidity. It kind of prevents the phospholipid tails from moving around. It kind of acts as a structural barrier. And it has a temperature component as well. So, for example, at high temperatures, it makes the membrane less fluid. So there's not going to be all moving around. If you think when normally you would heat things, that, the energy is going to make all the molecules float all around, cholesterol kind of helps resist that change. So it prevents fluidity at high temperatures, and at low temperatures, it prevents them from getting super closely packed. So overall, I would say that cholesterol is kind of acting as a general stabilizer. That's why it's saying that it resists change. It prevents it from getting too crazy at high temps and prevents it essentially from solidifying at low temps. So in terms of life adaptation, bacteria that live in crazy hot springs have unusual lipids. So they, for example, might have a ton of cholesterol in their membrane. Other species, like winter wheat, for example, is able to survive in cold temperatures. So they actually have an increase in unsaturated phospholipids as kind of an evolutionary adaptation so that they can have a higher fluidity in those cold temperatures. Okay, so I mentioned these two before. Too far. Integral just means integrated. So it is embedded or inserted into the membrane. It's transmembrane, meaning that it goes across the entire membrane through the tails and heads of the phospholipids. Peripheral just means that it's on one side, it's on the periphery. And it's able to be held in place by either the cytoskeleton, so that's the structural component inside the cell, or it's held in place by the ECM, which stands for extracellular matrix. So that's the structural component on the outside of the cell. And generally what they're doing is providing a stronger framework or a stronger structure. Things can be anchored to it if it's on the inside or the outside. So it gives that overall sense of structural integrity. So you can see here, as I kind of showed you before, the transmembrane or integral ones are the bigger, fatter ones that go all the way across. And then there are peripheral ones either on the inside or the outside. Here's what it looks like if you actually zoom in on one of those integral membrane proteins. You can see that it's got a series of alpha helix in the middle. And we had talked about that before in terms of proteins, where they have the primary structure and then they have the secondary structure, which alpha helix is one of them. And then based on that, they fold up into a tertiary structure. So that would be what you are seeing here. And all proteins have an amino N terminus or end, and they also have the carboxyl end, the C terminus. For whatever reason and forevermore, we always consider the N terminus is like the start and the C terminus is like the end. You can see here in the center would be where the hydrophobic amino acids like to live and where they folded themselves because it's a hydrophobic environment. The hydrophilic ones are going to want to have oriented themselves towards the water, whether it be on the extracellular side or the cytoplasmic side. Some of the general functions of what these things do. So one of the hallmarks is that they're going to act in transport. So whether it's ending up just being a passageway for certain molecules or needing energy to fuel this process, we'll learn about that later. These proteins also can act as enzymes. When they come in contact with a substrate, then they can act on it. They are also involved in signal transduction. So when there's molecules outside of the cell that are meant to trigger a response, they can bind to these proteins and then trigger a downstream effect, and we'll learn about that too. A lot of times it could be cell-to-cell recognition. So I always think of like cell surface markers on the outside of cells. So if this was like a bacteria of some kind, your bodies can recognize the surface markers and say, hey, that's foreign. We're going to attack that and get rid of it. That would be an example of cell-to-cell recognition. It's possible to do intercellular joining, so if they're going to stick together for some reason. And it also acts as a scaffold or an attachment point to the extracellular matrix. So it can be kind of anchor of that structural component to give it more integrity. Okay, carbohydrates. So their job is cell recognition. And they're used a lot in developing organisms. But you can see they're shown as little hexagons because most carbohydrates are 5 or 6 ring sugars. And so they're just kind of identifying markers. They're kind of like the fingerprints of the cell. Not all cell types and cell groups have the same carbohydrates or same structures on their surface. And so that can identify for your body one type versus another. These things can include glycolipids, as well as glycoproteins. In this slide, it's giving you the example of blood transfusions. So again, different blood cells have different cell markers. So A-type blood has type A carbohydrates on the surface, type B has type B carbohydrates on the surface, and that kind of thing. Okay, now we're going to shift over to membrane permeability. And I touched on it a little bit, but the idea here is that the membrane is a barrier. And it's, keeping the inside stuff in and the outside stuff out. But it is allowing certain things to leave and certain things like nutrients to come in. What can go through and what can't? Well, small nonpolar molecules have no problem going through because the core of the membrane is hydrophobic or nonpolar. So these guys are happy at home there, but they're small enough to kind of slip through the polar part of the membrane. So this includes things like nitrogen, carbon dioxide, oxygen. Those just go right on through. Small polar uncharged molecules can cross in small amounts. So what does this mean? Small polar uncharged molecules. Overall, we've learned that water is a polar molecule, but it only has like a tiny bit of a slightly positive end and a negative end, but overall, it's not charged. There's a balance of charges there. There's not a plus or a minus overall on the whole molecule, so that's why it's considered that. And in small amounts, it's able to pass through too. The hydrophobic nature of the internal portion of the membrane is able to effectively prevent things like ions, which are charged, or large polar molecules from actually going through. They're going to need to use helpers to make that happen if they need to get through. So either special channel proteins that are integrated in or special transport proteins to help them get through. So this last slide here is just talking about plant cell permeability because they have an additional layer. They have more than just a membrane. They also have a cell wall. So you can kind of see in this particular picture, the bottom layer is the plasma membrane. And then it has this thick, complex structure of kind of cellulose that is the cell wall. And that wall is basically an additional structural component. So it's an extra block between the inside of the cell and the outside of the cell. And these cell walls, just as their composition, are made of cellulose. And hopefully you remember from the macromolecule section that cellulose is a complex form of carbohydrate. So that comes together to kind of make that. And again, it gives it structural integrity to plants, but that's where it comes in. So overall, that's it for membranes.